what cats don't allow us to do is have those nicely, the diversity of dogs. First of all, you don't have the cats, and then it's not parceled so neatly into discrete populations. There are some, but it's not the same dynamic. So while there's some interesting things in the cats, it's not as easy to access them as in the dog. And frankly, there aren't as many things. I mean, one of the fascinating questions is, why has the dog changed so much and the cat changed so little? Is it simply a dynamic of human intervention, or is there something about the biology of those two members that makes the cat resistant to change and allows the dog to be highly flexible and tolerant? And I think it's successful for a lot of people. But there are clearly things in the cat that are of interest. Metabolism, diabetes, for example, kidney disease, and they also suffer from lymphoma. And there's a lot of aging variation. Yes.
From our hairless dog.
Yes.
And what did that give you over the rest of the
Okay, so her question was, so this was one of the left field, very generous
This
field.
is the clean hairless.
Yes, yes. So we were able, on a small number of dogs, we got not a skin biopsy sample that allowed us to look at the expression of genes, and I think this is probably what Karen was talking about before, the expression of genes in the different types of dogs, right? The carrier, the hairless, the hairy hairless, and the rat hairless. And what we found from that was that the stem cell markers were expressed like we thought. Were expressed, and we didn't think that they would be assigned. There was one gene that was very high in the hairless dogs, and then we found out that it was a gene that also responded to UV, ultraviolet light, so it was essentially just the sunlight. But what we were looking for, we were trying to use that to nail down the extent to which the stem cells were included. So that'll be in the paper. So it was very important.
You spoke about that in certain ways. Was the geometry study for its areas of interest?
So the question was deafness in Dalmatians, and what's really been clear for 50 years, mainly from mouse work, is that the genes that influence white spotting also contribute to a risk for deafness. And the reason for that is it turns out that the cells that produce pigment need to migrate early in development and cover the surface area. And when you have a dog that's white spotted, that's a region where those cells did not get to, right? So they can't produce pigment, so you have the white spot. There's one part of the inner ear called the stria vascularis, where those same cells need to get there in order to hear. If they don't make it there, then it can be unilateral or bilateral deafness. That's a congenital deafness, a developmental deafness that's tied to the extreme white in the case of Dalmatians. What no one yet understands is there's plenty of breeds that are extreme white that don't have the degree of deafness that the Dalmatian has. Yeah. And any breed that has white will have some frequency of congenital deafness. What we map is all three of those are very different from that. But no one yet understands that question. Why is the Dalmatian at such greater risk than, let's say, an English pointer? Yeah, so most of the mutations for white spotting have been identified. But this is the difference though between a disease that's determined by genes and a disease where your probability increases with genes. So any dog that has white probably has more or less the same probability. I should say that differently. The amount of white is what defines that probability, but then it's still a probability. So it's a little bit trickier from a breeding standpoint.
It is. But there is evidence associated with high voltage.
Right, exactly.
But there would be a different market that we could use.
There is, but most people you could just breed based on the amount of white that you see. Like if you wanted to get rid of that deafness, you just get rid of the white spot. Does that make sense?
But we have a 5-ball breed right today.
Right. It's the kind of trade-off. So I'll give you another example for this. So it now turns out that some of you may know that Dalmatians are also at risk for stone formation, uric acid stones. It's now clear that every single Dalmatian is fixed for that susceptibility. Not all of them throw stones, but genetically they're identical with respect to the risk of throwing stones. And it turns, so then you say, well, why is every Dalmatian at risk for this equally? It turns out that the gene for this probably makes the black circles better. So it's been under selection. So this is another thing. There's a lot of things that we select for that bring with it directly a risk for something else. And what people would, I mean, at some point it becomes clear, you know, it's a trade-off. And
That's why they bred the pointers to try to eliminate that gene with that.
right, and now people in the breed though are saying that that's not a purebred Dalmatian because it's got a pointer.
My question is by breeding animals that have like certain neurology problems in our breeders coming up like with ataxia and stuff, and we have been told not to totally discreet the positive dogs, that you can breed positive and negative. Are you breeding, like say if I was breeding a positive and negative and I took only the positive from that litter, and then I went into another litter and did the same thing where I had a positive and negative parents, and breeding two positive dogs together, am I doubling my chances or am I diluting that effective gene?
So it entirely depends on the basis of that ataxia. If it's recessive, if it's a simple recessive, then what that means is without a DNA test to tell you who's a carrier and who isn't, it becomes very difficult to know what you're going to get. What I would say though is now with these technologies, if we can get just 6 affected dogs, literally in 2 weeks, 6, and Erin's going to kill me when she hears this, 6 affected dogs in 2 weeks, we can get it back. And then what that allows you to do is look at, right, because litter mates, let's say you had 6 pups in the litter, and you mated a carrier to a non-carrier. On average, half of those puppies are going to carry it, and half are going to be clear. But it could be every pup in the litter is clear, or it could be every pup in the litter is a carrier. So once we have a signature, then you go in with a cheat swab and say, aha, these puppies are clear. Basically, but the problem is that ataxia could be multiple genes. It could be more complex. And so then to answer the question, it becomes very difficult. You just don't know. So it really depends on the inherent basis of the defect. But what I will say is I think for years, people in my field have said, have made this sound simple. Right? So if the dogs from a bloodline that produced epilepsy stop breeding, and that